Hello, my name is Enzo Perez. Today I will be discussing the process of meiosis. Meiosis is a division process that occurs in germline cells. It begins with a diploid cell. The diploid cell proceeds to divide into haploid gametes, which contain a singular copy of each chromosome. The haploid gametes that are produced as a result of meiosis could go on to produce offspring via sexual reproduction. Similarly to mitosis, the cells pass through interphase G1, S, and G2 stages before entering meiosis. During the S phase, DNA inside of germline cells are duplicated before meiosis commences. Meiosis is composed of two cell division events, which are meiosis 1 and meiosis 2, compared to how mitosis only has one division event. In meiosis 1, sister chromatids, or two halves of a duplicated chromosome, are separated. In mitosis 2, homologous chromosomes, which are similar chromosomes an organism receives from its parents, are separated as well. The process of mitosis begins after DNA is duplicated in the S phase.
The first phase of meiosis 1 is prophase 1, where DNA condenses to form chromosomes. Firstly, each pair of homologous chromosomes undergoes synapsis, which pairs two pairs of sister chromatids. After synapsis, recombination or crossing over takes place. Crossing over is the process in which homologous chromosomes trade parts with each other, ruining the identicality of the sister chromatids. Because crossing over occurs in completely random areas of the chromosomes, genetic diversity is increased. Lastly, the nuclear membrane commences breaking down as the centrosomes migrate to opposite ends of the cell and form microtubules, which attach to the chromosomes. The next phase of meiosis is metaphase 1, the phase in which the homologous pair of chromosomes line up at the metaphase plate. When they line up, they prepare for separation. However, the chromosomes align randomly, which allows gametes to have distinct sets of homologues, proving to be another opportunity in which genetic diversity can be increased. Following metaphase 1 is anaphase 1. During anaphase 1, the homologous chromosomes are separated, being pulled towards opposite ends of the cell. However, the sister chromatids remain attached to each other, which causes it to be necessary for another round of division through meiosis. Following anaphase 1 is telophase 1 and cytokinesis, which usually happen at the same time. Telophase 1 can be indicated when the chromosomes arrive at opposite poles of the cell, the sister chromatids still attached. The cell proceeds to divide into two haploid daughter cells. With the formation of two new daughter cells, the daughter cells must proceed to undergo meiosis 2. The new daughter cells do not replicate DNA. The first phase of meiosis 2 is prophase 2, which, similarly to prophase 1, facilitates the condensing of chromosomes and the breakdown of the nuclear envelope. The centrosomes move to opposite sides of the cell, and the spindles begin to form, attaching to the chromosomes. There is no synapsis, however, because the daughter cells each only contain one copy of each homologous chromosome. After prophase 2, metaphase 2 facilitates the chromosomes lining up along the metaphase plate. Once again, the chromosomes align at random, increasing the genetic diversity due to many possible combinations and outcomes from the random assortment of chromosomes. Following metaphase 2 is anaphase 2, where the sister chromatids are pulled towards opposite poles of the cell. After anaphase 2 is telophase 2 and cytokinesis, where the nuclear membrane reforms around the chromosomes. Additionally, the chromosome decondenses. The two daughter cells that were as a result of meiosis 1 both undergo cytokinesis, splitting both of the cells into haploid cells with only one chromatid. In total, 4 haploid cells are a result of the entire process of meiosis.
Even though mitosis and meiosis are different functions, they both share similarities and differences. To begin with, meiosis pairs homologous chromosomes in prophase 1. allowing crossing over. However, mitosis does not pair homologous chromosomes in its stage of prophase, leading to individual chromosomes lining up at the equatorial plate instead of homologous pairs aligning at the equatorial plate as seen in meiosis during metaphase. In anaphase, between meiosis and mitosis, centromeres separate in the anaphase of mitosis. However, in anaphase 1 of meiosis, the centromeres do not separate. Instead, the homologous chromosomes separate. Finally, the genetically distinct daughter cells of meiosis must undergo the procedure of meiosis 2, compared to mitosis, in which the daughter cells are genetically identical and do not need to go through another procedure. The final products of mitosis are 2 genetically identical daughter cells, while the products of meiosis are 4 haploid daughter cells that are genetically distinct. Meiosis is a fascinating process that contains a plethora of steps but ensures the successful production of gametes.